Hi everyone, and welcome to our video on epistasis. What we're going to be doing in this video is carrying on with our module 6 part 2 work, looking at all these different patterns of inheritance. And in this particular video we'll have a look at specification references 6.1.2b part 1, genetic diagrams to show those patterns of inheritance, and 6.1.2b part 2, the use of phenotypic ratios to identify epistasis, including the explanations about what epistasis actually is. I think the first thing that we really need to do before we launch into loads of fun Punnett squares as always is to understand what we mean by this phrase epistasis. Now, quite simply, the definition is that we've got these two non-linked loci that are going to be interacting so that one is going to mask the expression of another. So what we're really seeing here is two gene loci that aren't actually linked together that do have an influence on each other. So that's the key thing with our epistasis. We've got these non-linked loci that will influence each other in some way. And these genes may be antagonistic or they could be complementary in their action. So we've got those two different options and we'll have a look at each of them with some specific examples in this video. Now, because these gene loci are not linked, what we need to bear in mind is that that does mean that when it comes to the formation of our gametes, then they will be assorted independently. Because where they're not linked together, obviously random assortment at each of those stages of our process of formation of the gametes in meiosis can lead to that independent assortment. We get different variations within those gametes. When it comes to actually thinking about the genetic diversity and the impact of epistasis, then what we're going to see here is actually a reduction in genetic diversity. And the reason that we get that reduction in genetic diversity when epistasis has a part to play is because when we're considering our F2 generation, then because we're seeing a masking of an expression of a particular phenotype, then what we see are fewer phenotypes being expressed. Now, if we've got fewer phenotypes being expressed, obviously that can have an impact on the organism and its ability to survive in a given environment, etc. And therefore, we can get that reduction in the genetic diversity of that particular population. Okay, so what we're going to do then is we're going to start with the antagonistic epistasis first of all. And this is divided up into two different subcategories of antagonistic epistasis. So we're going to start with recessive antagonistic epistasis. When we're talking about this recessive epistasis then, what we're looking at is the presence of two recessive alleles at that first gene locus is then going to prevent the expression of whatever alleles may exist at the second locus. So what we're really seeing here is this masking of that second characteristic based on the homozygous recessive nature of that first gene locus. Now, obviously it's got to be homozygous because when we're talking about a recessive characteristic, the only way that we would see that impact is if we had both recessive alleles being present. So we must be homozygous at that first locus for the recessive characteristic there, and then that is going to prevent the expression of whatever alleles may be present at the second locus. So the way we could put this into fancy A-level language is that the alleles that are present at that first gene locus are epistatic to those at the second locus. And whatever alleles we have at the second locus are what we describe as hypostatic. So epistatic ones, the ones carrying out this masking, the hypostatic ones, the ones that are basically being masked. Now, what that looks like in terms of our very familiar capital and lowercase letters that we use within our genetic cross diagrams is we would be looking for those two lowercase letters, okay, our recessive alleles homozygous in their nature. Now, if that first gene locus is homozygous for the recessive allele, and I've given you the example of the AA there, then neither of the second gene potential combinations could be expressed. So whether we had a dominant and a recessive, or the two dominant or the recessives, it really makes no difference what we have at that second gene locus it will not be expressed because we've got that homozygous recessive position on that first. So in order for us to see some kind of outcome of that B locus in this case, we would have to have a dominant A. Okay, so we'd need a capital at that first gene locus. I think sometimes it's a bit easier to actually get this into your head once we've gone through a few examples. So we've actually got a chance to visualize what's happening here. Now, as always, we're going to go back to our old friends, the Punnett squares here. And my first example for you with our antagonistic and that recessive trait is going to be the salvia plants. Now, salvia plants are possibly flowers that you have growing in your garden. They're quite common in the UK. And what we find is they have basically our 3 different colours. So we'll have purple flowers, pink flowers, or white flowers, depending on which particular alleles we actually have present. So we're going to start off in our nice little original parent types with our pure breeding pink salvia. Now the pink salvias will have 2 capital A's and then 2 lowercase b's. So we're homozygous dominant for the A and we're homozygous recessive for the B. And we're going to cross those with pure breeding white salvias, which are homozygous recessive for A and homozygous dominant for B. So if we just work out what this would be in our simple little Punnett square, because we're starting off with pure breeding, we don't need any more than our 4 square Punnett. So what we've got, obviously, if we're talking about our pure breeding pink salvia first of all, then in each case our gametes are going to be a capital A and a lowercase b. If we then consider our pure breeding white salvias, in each case we are going to have a lowercase a and a capital B. So we've set up, obviously, the individual gametes that could be inherited. Then all we've got to do is fill out the squares beneath. So as always, whatever is in the top box, we fill out in the ones beneath, and whatever is to the side, fill out in the side. Now, there's no hard and fast rule on how you have to do this, but it's just a bit easier if you start with capitals and then put lowercase. It makes it easier on a quick glance to see what you're doing. So here we've got a capital A, so we're going to put that in first, and then we've got a lowercase a. Capital B on the left, and the lowercase b at the top. And just do that for all 4 boxes. So I'm going to just work down here. So we've got a capital A, lowercase a, capital B, and lowercase b. And then on the right, capital A, capital B, lowercase b, and the same again on that last box. So in our F1 generation, we have the exact same combination of alleles as the outcome. Now, because we've got a capital A and a capital B present within that final genotype in each case, all of these are going to be purple plants. Okay, so they're all going to be purple flowers in that case. So that's our F1 generation, but what we're really interested in is when we interbreed the F1 generation itself to get our F2 generation. Now, obviously we know our F1 generation in all cases there are capital A, lowercase a, capital B, lowercase b. So we need to work out the individual gametes that could form. Now this is where we're going to go to our lovely 16 square Punnett. So we need to put in our potential gametes first of all for both of our parents. The good news is they're both the same in this case because we're obviously starting with the F1 generation and they were all capital and lowercase for both A and B. So what we're going to have is a capital A, capital B. We're going to have a capital A, lowercase b. Then we're going to have a lowercase a, capital B, and a lowercase a, lowercase b. So these are going to be our original gametes that we start off with. I'll just quickly fill those in to the other ones. There we go. Now, same thing applies. It doesn't really matter how many of these squares we've got. Follow the same rules. Whatever is at the top, fill in the squares beneath. Whatever's to the left, fill into the squares on the right. So in that first one, we're going to have 2 capital A's, 2 capital B's. Then we're going to work across here. We've got 2 capital A's, a capital B, and a lowercase b. Then our third across, we are going to have a capital A, a lowercase a, and 2 capital B's. Then we've got capital A, lowercase a, capital B, lowercase b. You're kind of getting the idea here. all you're going to be doing is just working your way along very logically. Just take your time on this if you get it as an exam question, just so you don't make a silly mistake, okay? Because it's very easy, particularly in an A-level exam situation, when you are going to be a little bit more stressed because, you know, it's A-levels, they matter. Then don't rush it, okay? Because rushing is where you make silly mistakes. Now, another point to make is when you're actually writing your letters, please make sure that it is nice and clear what your capitals and your lowercase letters are, because some handwriting, it's not the easiest to unpick. So just take that little moment, do it properly, just so that you're not going to cost yourself a mark through either scruffy handwriting or carelessness, okay? So pretty much there now. Okay, so we've got all our different genotypes now filled in. What we then need to do is obviously work out our colours. Now, what we have said is that if we have got, obviously, the capital A and a capital B present, any number of them, we are going to have our purple plants. So I'm just going to put crosses in these in a purple colour just for simplicity. So any that have got a capital A and a capital B, these are going to be our purple plants. So we've got purple, purple, that one's going to be purple, another purple here, no capital B, so we can't have purple in that box. We can have a purple here, but not there. That one's purple, this one's purple, and then down here. There we go. So we've got all our purples marked off. Then we're going to go for the white ones. Now, obviously I can't use white, so I'm going to just have to put a W in the boxes for white, okay? Now, if we're looking for a white plant, that is going to be one where we have that homozygous recessive at the position for A, okay, so our first gene locus. So in that case, any that we've got, those two lowercase a's are going to be white. So we've got a white one, white, and a white one. What we do have are these three boxes left over, and I don't have a handy pink. I'm going to have to put a P in red, I'm afraid. I don't have a pink colour on my little colour palette. So in terms of our pink plants, these are going to be where we have a dominant in the A, so at first gene locus, but the second gene locus, the B gene locus, is recessive. So this one is going to be pink, this one here will be pink, and this last one is pink. So what we can see is we've got those three different colour combinations, and then if we count up the number of each, so let's start with our purples. We've got 4 on the top row, 2 on the next, that gives us 6, then another 2 is 8, and 1 at the bottom. So we have 9 purple, 2, 3 pinks, to 4 white. Now what we actually have here is that modified dihybrid inheritance ratio. If you remember, your dihybrid ratio was 9, 3, 3, 1. Now what we're seeing here, because of this epistasis effect, then this is actually being masked. So rather than having a 3 and a 1, we're ending up with those two going together to give us the 4. So when we're looking at this effect of epistasis, we're going to get a modified ratio, okay? So make sure that obviously you can fill out the Punnett squares, make sure that you are thinking very carefully about what's masking what. You'll get information in the questions help you with this. You're not expected to memorise any number of strange possible scenarios for weird, wonderful plants and animals, for example. Information will be in the question to help you. Just read it carefully. If it helps you to highlight, make notes at the side, do it, okay? And then just set out your Punnett square, work it out nice and carefully, and then you can work out your ratio. Now, the second type of epistasis we're going to look at is the dominant antagonistic epistasis. Now, in the dominant epistasis, if we have a dominant allele present for our epistatic gene, then we are not going to express the hypostatic gene. So again, what we're looking at here is what's present in that first gene locus. That is then going to determine whether the second is going to be expressed or not. So when we're thinking about this dominant epistasis example, we're going to have a look at chickens and the colour of their feathers. Because it's dominant epistasis, what we are going to need is only one of those capital I's, so a single dominant allele at that first gene locus, and that will then prevent the formation of colour in those feathers. So because obviously at that first gene locus we have two alleles, if it is either one capital I, one lowercase I, or they're both capital I's, we will not get colour. So basically we would end up with white chickens at that point. Now, in terms of what we're going to do for our little genetic breeding experiment then, we're going to start off with our pure breeding white chickens. Now, there's two ways that we can end up with white chickens because, you know, it's just fun like that. We could either have our capital I's and then capital C's. So in that case, because we've got obviously a dominant allele in the first gene locus, then we're not getting anything in the second anyway. Okay, so we've got our homozygous dominant in both of those gene loci. However, we could also have white chickens when we've got homozygous recessive in the first gene loci, so two lowercase i's, but there is also no pigmentation at the second. We've got the homozygous recessive for the C's. Okay, so in both of those cases we've got white chickens, but they're two different genotypes that could form them. So what we're going to do is we're going to start off by interbreeding our original pure breeding white chickens. That's going to give us our F1 generation, which is going to be heterozygous for both of those loci. So if we want to then work out what our F2 generation is going to be, we're going to start with our F1 generation, set out our Punnett square. It'll be the 16 again, because what we've got are obviously those 4 different gamete combinations that we could have. And then we are going to fill out our little Punnett square just as we did before. So we're going to start off with our capitals, and then we're going to fill in our lowercase, and just make sure that, as I said, you take your time. Now you'll notice how blatantly obvious I have tried to make the difference in my capital and lowercase c's, because c's are a little bit interesting. When you're doing these quite quickly, you can end up in a situation whereby you're writing it and it's not obvious what's a capital and what's a lowercase. Now, I always recommend that what you do is you really do exaggerate it. Notice how much bigger my capital C is than my lowercase. And the reason I'm doing that is it makes it far easier for an examiner to then spot when you've actually got a capital and lowercase if it is very different. If what you're doing is a little bit ambiguous, then if there's a requirement that we need to actually suss out where you've gone wrong in your actual Punnett square, where there could be a mark maybe for working that you haven't got the right answer at the end, but maybe you've got something further up, having it so explicit is going to make it much easier for us to see. So do make sure that you are making it really clear where there are those letters that it's not always as clear as like lowercase and capital I's. That's quite obvious. Okay, so as always, we're just going to go through and fill these in. And again, when you're doing this on your exam paper, do take your time so that you don't make mistakes. Okay, I'm trying to make sure I don't make any mistakes while talking to you as I do it. So let me just get these done very quickly. I see capital I, lowercase, lowercase. Then we've got lowercase, lowercase, capital, lowercase, and we've then got lowercase, 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 lowercase. There we go. Okay, so we then have our genotypes all filled in. Now, remember, this is if it's got a dominant allele at that first gene locus, so the I locus, then we're going to have white chickens. So we can start off by finding anything with a capital I, and we're going to identify them as being white. So literally anything that we can see with a capital I, you don't even have to look at the C's, it's just going to be a white chicken. So that's capital, capital there, and everything else is lowercase in that corner. Okay, now once we've done that, we then need to work out, are there any other potential white chickens? Because remember what we said in our original pure breeding generation, then we did have that scenario where we had white chickens that were homozygous recessive for both of those traits. So down in this bottom corner, we've got two lowercase i's, two lowercase c's, so there's another white chicken at the bottom. Then we've got these three where we've got, obviously, our I locus is recessive. So we've got homozygous recessive for our first locus, therefore we're not preventing colour, and we do have obviously a dominant or two dominants in the C locus, the second one. So in this case they are going to have some kind of colour. So these are going to be coloured chickens, as will that one, and this last one will have colour feathers too. Okay, so in this scenario, if you actually add them all up, then what we have are 13 white chickens to 3 coloured chickens. Okay, and again, if we're thinking about our ratio, we start off with our 9:3:1 ratio. What we're seeing in this case, because it's a dominant trait that we're seeing, this dominant epistasis, then we're merging this and this. So what we're seeing here is 9 plus 3 plus 1, that's obviously going to be 12, plus 1 is 13, gives us our 13 to 3 ratio. So it is just another example of this modified dihybrid ratio. The third type of epistasis we need to be aware about is the complementary. Now, what we typically see here is where we've got genes that are going to code for 2 enzymes that work in succession. So obviously each enzyme has its own gene, and these enzymes are then going to obviously work to give us some end result in a phenotype. Now, the example we're going to look at are some mice and their different coat colours, because mice are quite cute, as we'll see in my next little diagram. Okay. What we're going to consider here then is, if we think about our first gene locus, we're going to use the C's. Okay, so capital C, lowercase c, that's going to determine if the coat has colour, full stop. Okay, so that one's going to tell us, will this be a coloured coat? Then at the second locus, which we've got A's, this is going to determine what colour it's going to be. And the way it does that is actually by deciding the way that the pigment is distributed within those hair fibres. So we've got these 2 different gene loci that are going to then work together, so it's a complementary feature, to give us that final coat colour of our little mouse. So here's our cute mice, at least I think they're cute. What we end up with at the beginning is this colourless precursor. So that is our starting point, if you like. Now, if we end up with a dominant C, okay, so that's our first locus, this is then going to mean we get pigment. Now, if that was it alone, we'd just have black mice or white mice. That would be it. So if they had the dominant C, we'd have black mice. If they didn't, they would be colourless, or the albinos. But that's not it. There is this second gene locus that comes into play, which we've termed A. Now, if they've got the dominant A, that then distributes that pigment slightly differently within the fibre. So we end up with what we call an agouti pattern. And basically these ones have black pigments with yellow bands, but when you look at them, they kind of look like brown mice, in all honesty. So what we end up with are, if we've got mice where that first gene locus, the C's, are homozygous recessive, so both lowercase, we don't get a pigment, so they're albino or white mice. If, however, that C has a dominant allele present, we will have a coloured mouse. We know that. We just don't know what colour they will be at that point. Because to determine their final colour, we need to have a look at that second locus, the A locus. If we've got the dominant A, they will be agouti. If they're recessive for both of those alleles, they will be black. So let's have a look at what genotypes could actually exist in each of these different coat-coloured mice. Now, we're going to start with our little albino mouse here. Now, to be albino, they must be homozygous recessive for the C. Okay, so as we said, that C is determining whether that colourless precursor becomes a coloured pigment at all. So, because they're albino, they must be homozygous recessive. So in all of these potential combinations, we are going to have lowercase c's in every case. Now, because we've got this inability to change our colourless precursor to anything else, it doesn't really matter what's present at the A because it's blocked. So we could have an albino mouse that's got the dominant feature in the homozygous format. We could see it where it's heterozygous for A, or we could see it where it's homozygous recessive. The key thing there is that we've got the homozygous recessive at the position C, that first gene locus. If we then think about our black mice, so there they are in the middle, what we need here is at least one dominant C, so we need a capital C in there, okay? But they can't have a dominant allele for the agouti, so that means they're going to be homozygous recessive for A in each case. So our black mice would have to be AA, and then they must have a capital C, so they could be both capital C's, or they might be one capital, one lowercase c. They've got to have one capital C, they've got to have both lowercase a's. And then our last one, our little agouti mice then, these are the ones that have got to have at least one dominant for each of those gene loci. So we could see everything dominant. We could see both dominant A's and one dominant, one recessive for C. We could also see one dominant, one recessive A with both dominant C's, or we could have a dominant A, recessive A, dominant C, recessive C. So 4 potential genotypes for the agouti there. Key thing is we've got a dominant allele for each of those different gene loci. So again, we've got our Punnett square. We're going to be having a look at a 16 square Punnett square because we do have 4 potential of our gamete combinations that could be inherited. Okay, so again, all you're going to do is start off by filling it out nice and carefully, making sure that you don't make any errors as you go through. So whatever is at the top obviously goes into the boxes beneath, and whatever is to the left goes in the boxes to the side. So I'll try and do this nice and quickly. Let's see. Oh, that's terrible. See, that's where you've got to be careful when you're writing. Don't have like weird things that look like they might be C's when they're actually A's. Right, capital A, C. We've got capital, lowercase, 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 capital, lowercase, capital, lowercase. And then our final row, you'll be pleased to know, I've got that, we've got capital and lowercase, lowercase, capital, lowercase. Okay, so we've got our different genotypes all set up. Now we need to work out what colours our mice will be. So let's go back to what we said first of all. Let's start with our albinos. Now albino mice, remember, must be homozygous at the C locus. So we're looking for both lowercase c's, and when we find it, we're going to have our little albino. So I'm going to just call them albs, just for shorthand. So we've got another little albino there. We've got an albino here, and we've got an albino at the bottom. Okay, so we've identified our albino mice. Then we can think about our black mice. So black mice, remember, must be homozygous recessive for the A, but they must have at least one of the dominant C's. So we're going to have a black mouse here, we're going to have a black mouse there, and a black mouse there. Anything else is going to be an agouti mouse. Now I'm not going to sit and write them all in because we don't need to watch me just do that. But because everything else would be agouti, obviously if you're doing this as an exam question, please write in what they are. What we can then do is we can work out our little ratio. So what we've got in terms of our agouti, we've got 4 on the top, 5, 6, 7, 8, 9. So we've got 9 to 3 of our little black mice there to 4 of the white mice, the albinos. So again, we've got this modified ratio of that 9-3-3-1 dihybrid. So we've talked a lot about different ratios here, and one thing that you will have done over the past few lessons is you'll have had a look at even more ratios. So what I suggest you do as a good reference is just put them together. And what I've done for you here is I've just given you all those different ratios that you've encountered over this topic. So what we've done today is at the top, so we've got the recessive epistasis, a 9-3-4 ratio. Dominant epistasis, 2 different ways we could see that expressed, 12-3-1 or 13-3. And then we've got our complementary gene action, which actually has 3 different potential ratios, 9-7, 9-3-4 or 9-3-3-1. So what we have there are our different epistatic ratios. Remember them, okay? They're all modifications of the dihybrid inheritance. If we then just remind ourselves about previous ratios we've encountered, monogenic inheritance 3-1, dihybrid inheritance the 9-3-3-1, and then when you looked at codominance, that was the 1-2-1 ratio. So I recommend that what you do is just stick these on a flashcard, test yourself, make sure you know the different ratios for those different scenarios, because it can help you out with identifying what's happening if they give you a particular cross or combination of phenotypes that we can see and ask you what kind of inheritance it's demonstrating. These ratios will help you to do that. As always, I hope you found this video useful, and of course don't forget to subscribe to the channel so you can see when I next upload a video, and head on over to the website where you can find a range of resources to help you in your A-level biology studies.